Welcome to Study Loop.
Hi everyone, my name is Eric and let's go over problem 13. In problem 13, we are focusing on the 5S rRNA. So in order to answer the question, first let's define what this is. Now, as we can see, it is an RNA and this is located in eukaryotes. So this 5S rRNA or ribosomal RNA, that's what the R here stands for. This ribosomal RNA is a part of the ribosomal subunit. In specific, the large ribosomal subunit. Okay, so we have to remember that the ribosome subunit is made up of two parts. It's the large subunit along with the smaller subunit, right? So it's a complex that comes together, and this is what a ribosome would typically look like with a large and a small subunit. Okay, so the 5S rRNA is located in the large ribosomal subunit. And so the question is asking what is responsible for synthesizing the rRNA, specifically this 5S. And so let's go through the answer choices and eliminate those that are not relevant to this question. So we have four different choices. We have polymerase 1, polymerase 2, polymerase 3, and finally we have a ribonuclease 1. First off, let's start with answer D. Now notice how A, B, and C are polymerases, but ribonuclease is different. When you see nuclease, you have to think degrade. Okay? A nuclease its sole purpose is an enzyme that is going to degrade the bonds between a nucleic acid or a protein. Now we see ribo here. Ribo refers to protein. Therefore, a ribonuclease is going to be degrading protein. Therefore, if we're asking about a question of synthesizing, ribonuclease is not a relevant choice. Okay? Polymerases are those enzymes that are going to be synthesizing and putting together the bonds to make a biological molecule. So therefore, A, B, and C are relevant choices to pick from. However, we have to go over these three polymerases because they all make different things. Okay. Now these are going to be making rRNA. However, because there's three different versions, this is going to be something that you have to either look up and research or just memorize. So first off, polymerase 1 is typically going to make all of the rRNA in a eukaryotic cell, but it doesn't make 5S rRNA. So therefore, polymerase 1 is not an answer choice. In polymerase 2, it is going to make molecules such as the small nuclear RNAs. Okay, now these are going to be exonucleases that can actually break down molecules too. And these are not the RNA molecules we're looking for. So B is not the answer choice. And in C, polymerase 3, they're typically known to make the tRNAs or the transfer RNAs. Additionally, though, along with many other products, it is known to make the 5S rRNA. Therefore, our answer is going to be C, the polymerase 3, because it is known to make the 5S rRNA. I hope this helps explain these answer choices and how to quickly decide the answer. Thanks for watching and see you next time.